Good morning, everyone. Can you hear me, sir? Good afternoon, everyone. I am Chandaratri on behalf of Agrawal College, Balnavgarh. Welcome you all on the second day workshop on computer-aided drugs design. As you know that yesterday, Dr. Mukesh Global talked about drug properties and databases. I am sure that after attending yesterday's session, everyone is aware to the utmost importance of this in the today area, and especially in this era of pandemic utilization technology is the demand of the time. Now, I would like to invite the emeritus scientist, Professor Mukesh Dobal, to start the workshop. Over to you, sir.
Thank you very much. So I will continue on this. Are there any questions from yesterday's class? Did you all practice? Anybody has any questions, doubts? Did you practice some of the databases?
Yes, participants, do you have any query? Please come up with your queries.
Yeah. Did you practice yesterday at home? And did anybody have any doubts? Otherwise, we'll go forward.
Sir, at present, there is no query. You please continue.
Okay, I will continue. Okay, so we talked about why drug properties are very important. We talked about why drug properties are very important because yeah. Can you see my slide? Can you see my slide?
Yes, sir. Yes, sir.
Okay.
It's visible.
Okay, so drug properties are very, very important because most of the drugs fail because of lack of ADME, that is absorption, distribution, metabolism, excretion, lack of efficacy, that means bioavailability, toxicity. So all these reasons, so they all add up to 94, 95%. So whatever we develop in the lab will not go through because of these reasons. So we have to give a lot of importance to these properties, the properties of the molecules. That's very extremely important. Okay, there are so many properties. Logarithm of P. Then logarithm of P is the hydrophobic hydrophilic balance. Hydrophobic because the drug has to cross the GI. Hydrophilic because it has to be water soluble. And then things like we looked at many rules. One important rule is Lipinskis Rule of 5, which tells if you have these properties, chances are it will have good oral bioavailability, good oral absorption. Okay, we are talking oral because we consider only oral drugs. If it is intravenous, you can directly give it in the into the blood. Okay, so if any of these or more than one property does not satisfy, chances are it will not have good oral bioavailability. So if you are having a lot of new molecules and you don't know the properties, you can check it out from Lipinski's rule whether they follow the properties or not. Okay, I showed you yesterday Swiss ADME is one nice software. We can get the properties of any molecule, many properties. Lipinski's rule is one, and then there is something called the Gauss et al. Rule. Then there is something called Weber rule. Then there is something called Mugue et al. Rule. And then there is something, the total polar surface area related absorption rule, blood-brain barrier rule. Okay, and then P, glycoprotein efflux transportation related issues, and then OPRIA rule. So many rules are there. If you have a molecule, you can check it out using Swiss ADME, whether all the rules it satisfies. There are many softwares which can do. I showed you another software also, which can check out various properties. Okay. Which can another software, open source software is there, which can also measure the toxicity. I showed you that yesterday. Okay, that's called a PK, PDMET. And so we can calculate the properties. They are not measured. That means they are not experimentally determined. But if it is a drug, as I said, the drug means it's approved by a Food and Drug Administration. Then you can get properties of that from these databases: Drug Bank, drugs.com, FDA. These databases give you all the information about drugs. That means those which are being approved by FDA, which are there for certain treatment. Okay, you will get all the experimental data. But for many other things, you will not get experimental data. Maybe you have to look into literature. Maybe if you are a chemist and you are synthesizing new molecule and you want to know the properties, we can use Swiss ADME, or we can use a pre-ADMET toxicity, all those softwares to calculate these properties. Okay. And then if you think one property is not good, you can try to, as a chemist, modify that. For example, if it's very hydrophobic, it looks, that is log P is higher, then you may think of reducing log P by making it more hydrophilic, maybe add OH group or add NH group. Like that, you can do some modifications to the structure to improve something. If it is too much hydrophobic, it is not water soluble, then you may think of adding some OH groups, or you can make a salt out of it. Okay, if it is very hydrophilic and you think it will not have GA penetration, you may think about making it hydrophobic. Like that, you can think of modifying many things actually. Okay, so once you measure, once you calculate all these properties, these are all calculated properties. You have to remember that actually. Okay. So we saw a lot of things yesterday and looked at many databases yesterday, and we know how to apply these rules. And there is one very important technique which I talked about, that's called smiles. Yeah, smiles notation. Smiles is one way, the excellent way of representing text representing of molecules. Okay, so you can transfer structure information from one database to another. That's called smiles. Okay, it's a very useful way of text way of representing. We can represent single bond, double bond, triple bond, stereochemistry, aromatic rings, aliphatic chains. Everything could be represented, and then branching. Everything could be nicely represented in a string notation. So smiles is a very important notation for the whole thing actually. Okay. Okay. Let's look at some structures just to get okay, this is Humira. This is the commercial name. This is one of the top selling drugs for rheumatoid arthritis actually. It's a big business because, like I said yesterday, as the population ages, especially in the Western country, one of the major ailment is rheumatoid arthritis, swelling of the joints, pain, and so on. So this is an oral drug. Okay. This is the volume of distribution, 87 liters. Very interesting point you have to notice. The drug not only gets distributed in the blood, but if it is a hydrophobic molecule, it may even enter the tissues, it may enter the fats if it's very hydrophobic, if it is small molecule also. So the volume of distribution can be different for the same person. If it is a very hydrophilic, it will remain, and a large molecule like a protein, it may remain in only in the blood. Whereas if it's a small molecule or hydrophobic, it can distribute it not only in the blood, but also in the tissues and fats. So the volume of distribution changes quite a lot, very small to very large value. Why is it important? If the volume of distribution is very large, it is like putting 100 grams of salt in a bucket of water. It gets distributed and concentration comes down to amount of salt put divided by volume. Suppose if the volume of distribution is very large, the concentration will be very low, correct? So that's why volume of distribution is a very important parameter. So this is a small molecule. So you see 87 liters. So it's not only the blood volume, but also it may be diffusing into the tissues also. It binds 40% of the drug binds to the proteins present in your body actually, and bioavailability is very good, 74%. Okay, and elimination, it gets, as you know, any xenobiotics, any foreign compounds get eliminated from the body because of biotransformation in the liver and then urine excretion actually. So in 3 hours, half of the drug concentration gets eliminated. Okay, so it gives you metabolism. Like I said, for drugs, you have to, the pharma companies collect all the data actually. Okay, so they tell you how it gets metabolized. Like, there are many enzymes called cytochrome P450 present in the liver, which metabolizes any xenobiotics, any toxic chemicals to, there is an oxidation reaction, hydroxylation, hydrogenation reaction, and then there are sulfation type of reactions. Okay, so the compound becomes very hydrophilic so that it can be eliminated by urine actually. So there are many enzymes, P450 enzymes. So this is how it gets metabolized. Now, what else? Yeah, clearance within 70% through hepatic, renal, and so on. Now let's look at another drug. Look at this. This is again given for rheumatoid arthritis. It's a biological autoimmune disease. This is a large molecule, very large protein, as you can see. It is acting on the genes related to inflammation. Again, for rheumatoid arthritis, it contains 934 amino acids. So molecular weight is very high, as you can see the structure here. So what happens? This drug has to be given intravenous. It cannot be given orally. Number one. Number two, look at the half-life. It's much larger. 102. It's half-life 102 hours actually. When you compare to the previous one, very high. Because it's a protein, it also has an isoelectric pH. Okay, as I said, it is given as an injection under the skin. Okay, let's look at the next one. Another important drug. And again, this is a very big molecule. It's again used for autoimmune diseases and rheumatoid arthritis. Look at the formula, molecular mass. It's a big molecular mass. Half-life is 9.5 days. And then bioavailability is 9. Volume of distribution. Look at the volume. It's very low, 3 to 6 liters, because it's a large molecule. It's very hydrophilic. It will remain only in the blood. As you know, blood contains 70% water. It will not be able to go into tissues, fats, which is very hydrophobic. So the volume is so low. Whereas if you remember the previous case, volume of distribution is 87 liters. A small molecule, we find very high volume of distribution. So volume of distribution can change depending upon the size of the drug, depending upon the hydrophobic nature of the drug. Okay. Look at another one, another drug used to treat autoimmune disease and rheumatoid arthritis. Okay, so you can see it's got also a very high low volume of distribution. So depending upon the nature of the drug, it's either given intravenous or oral, and the volume of distribution changes a lot, and the half-life of the drug also changes a lot. Okay, and so on. So these are very important pharmacokinetic parameters, which helps one to decide the dosing strategy, which decide the rate, and so on. Okay, so far we talked about properties. Now let's look at something drug design. There are two types of designs. One is called the ligand-based design, other is called the target-based design. Ligand is your drug or the lead molecule which you are trying to design. Okay, that's why we call it ligand. Target means the target protein, okay, the target protein or target enzyme and so on. So that means you should be knowing details about the target actually. If you do not know the details about the target, you can do only this left-hand side, ligand-based design only. Only if you know the target details, then you will be able to do the right-hand side of it actually. Okay. Now, in ligand-based design, there are different approaches there actually. We have pharmacophore-based design, QSAR-based design, fingerprint-based design. Okay, I'm going to talk about pharmacophore-based design today and the QSAR-based design tomorrow. And in the target-based design, we have this docking approach. That means you are docking your ligand to your protein. That is what is target-based design is all about. Okay, you are docking your ligand to your protein. So the left-hand side is called the ligand-based design, where you look at only the small molecule, the lead molecule, and using pharmacophore-based approach or quantitative structure activity-based approach, you design new molecules. In the target-based, based on how your ligand goes and binds to your target, you try to design new molecules, okay, based on the binding efficiency, binding energy, binding affinity. Okay, ultimately you have to anyway check the filters, drug likeness property. You can't avoid this. Lipinskis rule, all the various rules, no, Muge rule, Egan rule, Opera rule of 3, blood-brain barrier, bioavailability. So we have to use all the rules to either filter out, either accept this for experimental studies or not accept this for experimental studies. Okay, so this filtering is always going to be there. You can't avoid that actually. Okay, so we start with ligand-based design, which is small molecules. You use a pharmacophore-based approach or a QSAR-based approach, okay, or like a fingerprint-based approach. In target-based design, you know details about your target protein into which your drug goes and binds to. So we use docking type of approach. And finally, whatever new molecules you design on the computer screen, we have to check whether the drug likeness property, ADME properties, toxicity properties, and select those which you think are good and reject those which you think are not good. Okay, then you go to your lab experimental studies. This is so this whole portion is called the computer assisted design of molecules. Okay. And as I have been talking in the past last day, this properties are very important. So you screen the molecules finally based on the properties before you go for experimental validation in vitro lab. Later on, you may go into animal studies, then you may go into human volunteer studies and so on. So the entire computer aided approach can be like this actually, the ligand based and target based. Okay. Now we will look at the ligand based, in which we have pharmacophore based approach. We will try to look at that more in detail actually. What is this pharmacophore? Yeah, pharmacophore is a description of molecular features which are necessary for the ligand to go and bind to a biological macromolecule. Macromolecule means big molecule like an enzyme or protein and so on actually. So there are some features which the molecule should have so that it can go and recognize a target protein. Okay, that's what is a pharmacophore. So if you look at IUPAC, they define pharmacophore as steric, electronic features that is necessary to ensure optimal supramolecular interaction with the target. Okay. So the diverse molecule may have same activity. So why does it so? It may look diverse, but there are some features which may be common, so that they are exhibiting a particular biological activity. Those features are called pharmacophore features. Those features are called pharmacophore. Okay, so structurally diverse ligands having common receptor to bind, common performance. Okay, so probably they are having common pharmacophore. What are these pharmacophores? Hydrophobic, aromatic rings, hydrogen bond acceptors, hydrogen bond donors, cations, anions. These are what is defined as pharmacophore. So if you have a 5 or 6 molecule, they all seem to have say antibacterial property towards E. coli. So I want to know what are their common features. What are the common pharmacophore? Do they have some hydrophobic groups common? Do they have some aromatic groups common, or do they have some hydrogen bond acceptors, donors common, and so on? So that is what we will try to find out. That is called the pharmacophore identification. Okay. So, for example, if you take basketball players, okay, you will see that the common feature could be all of them are tall, 6 feet tall, right? That could be a common feature. Of course, there could be shorter basketball players also, but generally one feature could be tall guys. Okay, so that is a pharmacophore feature, or maybe they have very long legs. That could be a pharmacophore feature, actually, right? If you look at say weightlifters, for example, they will have very thick arms, maybe thick biceps, okay, or thick or strong legs, thick legs. Okay, that could be the pharmacophore feature for maybe a weightlifter. So what's the advantage of this identifying pharmacophore? So the advantage is I can look into, say, zinc database. You know, zinc database contains 750 million compounds. So I can look into that database and see whether there are molecules with that feature. So that molecule may also exhibit same activity. So suppose I am looking at liver cancer, and there are 10 drugs reported in the literature for liver cancer. I try to find out what are the common pharmacophore features of the 10 molecules. Suppose I find some features, could be hydrophobic or aromatic or acceptors, hydrogen bond. Okay, then I know these features. I take and start looking into zinc database, for example, which contains 750 million, and see whether such features are there with some molecule. And suppose I identify a molecule with that feature, probably that will also have the same anti-cancer activity related to the liver. Maybe they also, those molecules also with those features have similar activity. That's the advantage of doing this type of pharmacophore based approach. For example, I said common pharmacophore feature for basketball player could be, say, tall people. Okay, so if I want to select basketball players, what will I do? I will go to a classroom and call all of them who are 6 feet tall, because the pharmacophore feature, common feature for a good basketball player is 6 feet. So hopefully they will also be good basketball players. Okay. Of course, they might not be, but at least they have those features. You can have 2-3 features: tall, 6 feet tall, maybe 3 feet long legs. So that could be 2-3 features like that. Similarly, for a weightlifter, maybe very big biceps, maybe big chest. Okay, those could be the pharmacophore features. So I go to classrooms and maybe pick up such people only. So I don't go to a classroom and pick up a very thin looking person. Chances are he might not be able to lift very heavy weights. So I will go for those who have big chest and maybe thick biceps. Those are the pharmacophore features. That's the advantage of doing this type of pharmacophore based modeling. We identify the features, common features of certain molecules which have exhibited certain activity. And then with those features, I can search databases and come up with new molecules with the same feature. Maybe even I can maybe get a patent. Maybe it's a new molecule if it is showing some experimental activity. That's a pharmacophore model works on. That's the principle of pharmacophore. So basically they look only at these: hydrophobic, aromatic, hydrogen bond acceptors, hydrogen bond donors, cations, anions. Okay. Okay. We will do it with an example. These are some drugs in the market. They are called selective COX-2 inhibitors. COX-2 is a cyclooxygenase enzyme. It's a prostaglandin synthase enzyme 2. This enzyme is there in the inflammatory pathway in arachidonic acid. It's called arachidonic acid or inflammatory pathway. And these drugs go and bind selectively to this enzyme. This is manufactured. You can see the company Pfizer, Merck, Pfizer. They call it valedicoxib. This is the commercial name. Roficoxib, celecoxib. Okay, this is a molecule still in clinical trial, so it does not have a name as such. It's called DUP697. So these 4 molecules, they selectively bind to the enzyme called cyclooxygenase 2 and inhibit the enzyme, and thereby progression of inflammation is stopped. So I want to know what are the common features of these 4 drugs. Okay, and as I said, if I identify some common pharmacophores, I can maybe look into databases to see whether there are any new molecules in zinc, for example, which have the same features. Okay, so it's a step-by-step approach. Many things you have to do to arrive at this. Okay, first you go to the zinc database and pick up one of the molecules, say Celebrex, celecoxib. You can download the file. It will be in SDF format only. But this pharmacophore search program requires in MOL2 format, MOL2 format. So we use another software called OpenBabble, okay, to convert the SDF format into MOL2 format. Okay. Then I go to Zinc and look for rofecoxib, download the SDF format, use OpenBabble, convert that into Malta format. Then I go to Zinc, look for validicoxib, download the SDF format, and then use OpenBabble, convert into Malta. Then I go to Zinc database, DUP697 maybe will be there. Download the SDF format, use OpenBabble, convert into Mol2. So all these 4 files will be in Mol2. Put them in a zip file, and then go to this online server called Pharmacist, and then upload your zip file. Then through your email, it will come out with the common pharmacophore, which you can view through PiMode. Okay, so we will start step by step. I hope you understood that. So each structure, go to Zinc, download the SDF format, convert into Mol2 format using OpenBabble, put all the 4 structures in zip file, go to this Pharmacist online server, upload your zip file, and run, and then results will come in your email, and you can view the common pharmacophore using PiMode. So you need OpenBabble and PiMode, and this is online server actually. And then there is another step for looking for new structures that we will see later. First is this portion. I hope you understood this. So we will start doing that. Any doubts? Anybody has doubts, please ask me.
Yes, participants.
Anybody
Till
has-
now, any query?
Okay. We will go step by step. Celebrex. So in the show, Celebrex. We'll look at. I'll go to Zinc database. Yeah, Celebrex. First molecule I'm trying to see. Silicoxib is called. Silicoxib. Can you see this silicoxib, all of you? Is it visible, silicoxib?
Yes, sir, it is visible.
Okay. So we want to download this structure. As I yesterday said, today also said, only SDF format is available, SDF. So we have to download this. I have downloaded that. So I go to download. Okay, so I'm saving it on the desktop just for fun. Okay, so far so good. Then we will go to the next molecule. Celebrex has been done. Rofecoxib. I hope yesterday you have been using Zinc well, so you must be conversant with that, right? All of you, I hope you have used. Rofecoxib is also there. Okay, so I hope you are quite conversant with Zinc and the Swiss ADME and all. So Rofecoxib also I will download. SDF format coming out. Okay, I will save it. Desktop. Okay, I've saved it. Next chap is validicoxib. Validicoxib. Yeah, validicoxib is here. Again, SDF format. Desktop. And then finally, you have DUP 697. This is still not a drug. That's why it does not have a nice name. It's still under clinical trials. Okay, so I have 4 of these files. I will save it in some folder. Silicoxide, DUP, ruticoxide, valericoxide. Good. Now I need to use OpenBabble to convert them into mol2 file. Okay, so I hope you all have downloaded OpenBabble. Okay, so what we do is I also have OpenBabble. This is a very useful program. It can convert any graphical file into any graphical file. You will be using it if you are going to be a serious CAD person. You will be using this OpenBabble quite a lot. It can convert many extensions into many extensions. Okay, see, look at this. So many different extensions are there. So here we can convert. Yeah, into any of this. So we can convert again into any form. Mol2 form. Okay, so SDF to mol2 form. I have to give the file. Silicoxib, open. Okay, so it will have converted multi format. So it has converted silicoxib into multi format. So like that, one after another, I do that. We have Duke also converted. And Doficoxib. Doficoxib also converted. And finally, Valedicoxib. So we have 4 files converted into MOL2 format. Valedicoxib, Doficoxib, Duke, Silicon. Understood? You can view these files using PiMode. We can view the files using PiMode. This is called PiMode. This is also a free software. MOL2 files can be viewed using PiMode. This is silicoxib. It doesn't put hydrogens. Normally, hydrogens are not put in unless we want specifically hydrogens. Just to see whether the structures have been properly converted into MOL2 format, you can use PiMode to view them. So far, everybody got it? Can you see the PiMode?
Yes, sir.
Yeah. It is good to see each structure separately because just want to see whether you downloaded the SDF format, converted into MOL2, just to see actually. This is another molecule. Yeah. So like that, all the 4 molecules you can see. see other molecule also. So this is then we have the vanadic oxide. So all the four molecules, we have the PiMode structures of all these four molecules. Okay, they all look good. You can confirm the structures also, whether they are all okay from the zinc database, whatever you got. So you're okay now. Now we can put them all in zip file. All the four molecules, we have them in zip file, right? So far, everybody understood this? Any doubt? Now we go to Pharmacist. So we did that up to this. Understood? Downloaded the SDF files from Zinc database, used OpenBabble, then converted that into mol2 format, took all the four, zip them together. Okay, now we are going to use Pharmacist. Yeah, this is Pharmacist. It's free for us, for everybody. Okay, it says give your email ID here. I give my email ID. Upload input molecular in mol2 format. Mol2 format. Choose file. Go to desktop. Okay, I've given the file, then email ID. So it will come to my email actually. Okay. Running Pharmacist, 4 molecules, results will be sent to this particular email ID. Okay, so generally it will take a few hours depending upon the load, 4-5 hours, then your results. So I had already run it yesterday. So results are already there in Zinc database. Sorry, in my email. Yeah, this is how the results come, pp doc. Okay, I have to click this. I had sent the link to Dr. Meena this morning. So I think she can share the link. If you click on the link, you will see the results. Dr.
Okay,
Meena,
sir.
can you share it with them? Yeah. Please share. All of you can click on the. This is how the results come.
Sir, you please share that link in chat box.
Chat box. Okay. Yeah. Now you can click on that and have a look at it. Our results will be there. Yeah. So it's now you can look at the results. This is how the results will come. Now we have to analyze the results. Okay. All of you saw the link?
Yes, sir. It's visible.
Yeah. Click on the link. You get the this page.
Participants, sir has shared the link in chat box.
Yeah, just click on it and you will see the.
Ah,
Everybody is getting. Just say yes or something. Just type yes so that I will know. Yeah, somebody type yes so that I will know. Yeah, good.
yes, yes, they are
Very nice.
trying.
And so these are the 4 molecules which I have given as input. I want to know the common features of these 4 molecules. So each molecule has some aromatic, hydrophobic, donors, acceptors, blah, blah, negatives or spatial features and all. Now these 4 molecules are superimposed in a 3-dimensional way. They are superimposed in a 3-dimensional way. And the common features of these 4 molecules will be seen. That's what this Pharmacist does. Normally it takes 3-4 hours. Because I ran it today morning. Okay, now look at this. It has aligned all the 4 molecules. It found 4 features common. 1 hydrophobic group, 3 acceptors. Acceptor means oxygen or nitrogen. 3 of them. 4 of them are common to all these 4 molecules, 4 features. And the score is 12.7. And there is another one, it gives 3 acceptors only. The score is giving line 11.9, like that, it gives you score. 4 features, 3 features, 3 features, and so on. And then as we go down, it will also give aligning 3 molecules at a time also, like that. If you have 5 molecules, it will do 5, 4, 3 also. So you can look at these numbers. So as you can see, it has aligned valid epoxy group, giving some scores. Sometimes if you align only 3, you may get a better comment. If you align more molecules, the common features may go down. If you align less molecules, the common features will go up. Okay, so here I have aligned all the 4 molecules, and there are 4 features that are common. 1 hydrophobic group, 3 acceptors, right? Now let's look at this. This is the highest score also. Let's look at this. All of you click on it. So it is giving 1 hydrophobic, 3 acceptors, and then you can download this file also. So we can say save link as. I can save it somewhere. Save. I have saved it in my this folder. Yeah. So this is my result of my pharmacophore superimposing all these 4 molecules. Now we can view this in PiMode. This is very important. We will use many other things. We will be doing many things with that. This is viewing through PiMode. PiMode. So what I will do is I will drag and bring it here. Yeah. So it has found 4 features, 3 acceptors and 1 hydrophobic, right? 3 acceptors, 1 hydrophobic. Now, which one is which? We'll find out. No problem. So, PiMode, please learn how to use PiMode. Spend some time. Like right click, I get. So this is a hydrophobic. First left click, then right click, go to label, go to atom name, it gives you acceptor. And then similarly go here, left click, then right click, go to label, atom name, acceptor, go to left click, right click, label, atom name, acceptor. So it's got the, see, you see 3 acceptors like a triangle and one hydrophobic group. 3 acceptors, and this is like an isosceles triangle. You know, these two sides look same. This acceptor is close. This acceptor is far away from these 3 acceptors, and there is a hydrophobic group. We can even get the distances of this. Go to wizard measurements. Okay, then type acceptor 7 angstroms. Acceptor 2.5 angstrom, acceptor 7.7 angstrom. So as I said, this is an isosceles triangle. Two sides are almost equal. One side is very close. So like a isosceles triangle, one acceptor is far away. Two acceptors are closer. Okay, then there is a hydrophobic group also. Okay, so you can get the distances. Yeah. So 3 acceptors and one hydrophobic is coming out for this particular system of anti-inflammatory. So if you look at these 4 molecules, they have 3 acceptors. Maybe look at this nitrogen. 2 nitrogens are closer, 1 oxygen is like an isosceles triangle. And then there is a hydrophobic group. 2 oxygen closer, 1 oxygen far away. Acceptors, right? Like an isosceles, there is a hydrophobic group. Then 1 nitrogen, oxygen, 1 oxygen. So this is like an isosceles triangle and 1 hydrophobic group. So that is the common, same thing with that. That is the common feature of these 4 molecules. 3 acceptors, 1, 2, 3, and 1 hydrophobic. Everybody is able to see this?
Yes, sir.
3 acceptors, 1 hydrophobic group. So this tries to superimpose each of the molecule and see where the acceptors and where are the hydrophobic groups and so on. So you can see 2 acceptors and 1 more acceptor here, right? And the hydrophobic group here. another molecule. So we can view each one of this molecule and see how it matches with the identified pharmacophore. Okay, so two oxygens here, one oxygen here. That is the acceptor, acceptor. So PiMode, you need to spend some time, look at those commands, measurement commands, uh, viewing commands. So if everyone has downloaded OpenBabble, you can practice this part. You can practice, go to Zinc database, get one structure out, get the SDF format, see whether you can convert into Mol2. Can you try this part? Because I have gave you the result. You are viewing with PiMode, but you should also be able to do this part. Can you just try it out now, all of you? Go to Zinc database. Each one of the molecule you download in SDF format. Use OpenBabble, convert into Mol2, zip it, go to Pharmacist and upload your zip file. And try it. Yes. So you can practice. We will wait for half an hour exactly. Anybody who has done the entire process, the first step, going up to uploading, that will be good because I have given you the result and you are able to view it in PiMode. But how to get the result is a main challenge actually. So we'll just give you 30 minutes, exactly 30 minutes. 4:30. Please tell me anybody has done it or if you have any doubts, you can put it in the chat.
Yes, sir. They can practice.
Yes, now itself, so that immediately I will know how to spell.
So we'll meet at 4:30, sir. Now.
Yeah, I'll be here. I am
Okay.
here. So in case anybody
Yes,
has doubts also.
we all are here.
Yeah, I am there. So in case there are any doubts.
Participants, you please practice, and then sir will resume the session at 4:30.
If you have doubts, you can just put it in the chat. I will try to see.
Yes, you can put your all queries in chat box. Hello, excuse me, sir. Excuse me, sir.
Was anybody able to upload the zip file?
Yes, participants.
Yeah,
any-
otherwise what is the
anyone?
point? We should be able to upload the zip file. So
Yes,
that,
yes. Have you tried?
so that if there are any issues, I can immediately help you out.
Sir, actually, they will practice, but I think everything was clear today.
No,
यहाँ
no,
तक तो
unless
clear
they
है, sir.
practice,
Yes.
otherwise it's not more
बिल्कुल.
like watching.
डॉक्टर सुखविंदर, आपका हुआ zip file आपने?
I go to home, they will have more doubts. That's why I
Yes.
thought it's good. Somebody has asked, may you show the procedure again? Again. That I can show, no problem. I can show that. That's not a problem. I'll show it. Did anybody do in the half an hour or just no? Because unless you practice, it's of no use. You may forget tomorrow. I'm telling you, in 2 days you'll forget the whole thing. You practice.
Actually, some participants have called me and they said that they will practice in late night. So actually, so maybe this 10-15 minutes was insufficient for them. So they will practice it surely, because it was very much clear, sir, today's lecture.
No, but then it doesn't work like that. Practice is different from seeing
correct.
things. Because
Has
when they
anyone,
they get stuck.
able to get the zip file?
When they get stuck, then they will give up because there is nobody to help. When we do it one-on-one live, when they get stuck, immediately we answer, so it becomes easy actually. Whereas when they do it at home, when they get stuck, they will be in big problem. Okay, so that's what this workshop is all about. So you have a one-to-one understanding. I'm telling you 100%. If you think I'll do it after 2 days, you will not do it because many things would have been forgotten. That's how it works actually. Because this contains a lot of steps, step-by-step approach. So
Participants are trying, sir.
Yeah. Unfortunately, it's not just like a lecture where you just watch and go away.
Yes.
Because if you at home, if you get stuck, then you will not be able to have anybody helping you. It will stop. So whereas in a workshop, we have an interaction. So somebody asked the question.
Some participants are sending message personally that they have not completed yet.
No, SDF file of Dextra is not downloading. Of course, I just downloaded. Okay, so I
They
don't
are
know what
able
it
to
is actually. So,
they are able up to do up to step 1 SDF format and
no, that is one.
mol 2 format भी कर लिया sir उन्होंने, but they have not completed up to this zip file and copy into the Pharmacist.
Because if you make a mistake in mol2 file, Pharmacist will give an error and it will not. So basically we have the SDF format, then mol2 format, convert into
Yes.
OpenBabble, and then we have to put them, upload in the Pharmacist. Somebody asked extra related. So don't call it Bextra, call it Validicoxib maybe, because Validicoxib is the this is Validicoxib. So Validicoxib you can download, no problem. So it's downloading. You can see somebody had a problem downloading Validicoxib. So it's downloading, no problem.
I think someone is asking after SDF format. SDF file of Bextra is not downloading. Okay.
Yeah, I have downloaded that, right? I have
Yes.
downloaded it. So you must have been doing some mistake. Please check. Call it Validicoxib because that's the name, chemical name. And then of course SDF format is this actually. So it gets downloaded here. And then of course I also mentioned about the OpenBabble. So OpenBabble, you give the SDF format and you will get the mol2 format, give the input file, output file, and then you convert. So that should come into the mol2 format actually. Okay, so for example, Celebrex, open, okay, then so convert, that's it. So it will get converted. So that's how you do all the mol2 files. Once you have all the mol2 files, you put it in the zip. All these 4 files in the zip. But I suggest you check it out on PiMode just to see whether those files are downloaded and converted files structures are okay. It I mentioned, right? So PiMode. Each of the file you please check just to see whether it's okay. There is another. So with the PiMode, you should know all the commands, how to find out what are the various pharmacophores it has found. For 3 acceptors, 1 hydrophobic group. So you should be able to get that. So how do you get that? You get the result through your email, and you will be able to see. Yeah, you will be able to see the results. It gives you all the features here, and you can select the highest score. And it says 3 acceptors, 1 hydrophobic group. Okay. So did anybody get this so far? Okay, were you able to upload the zip file? Anybody?
Yes, participants, please respond.
Yeah, you can.
Any one of you able
it in
to
chat.
upload zip file?
Okay, so the common features of this common pharmacophore features of these 4 molecules are 3 acceptors, like an isosceles triangle, and 1 hydrophobic groups. So 3 acceptors, hydrophobic group in between, 3 acceptors and 1 hydrophobic group, like that. 3 acceptors and a hydrophobic group. Okay, and that's the common feature of all these 4 molecules actually. So Pharmacist helps you to determine that based on the structures which you have given in a Molto format. So once you have this done, you can look at whether there are any other molecule in the databases with this type of feature. Okay. Using some other server called Zinc Farmer. Zinc Farmer gives you an idea. It will search the zinc database and try to find out molecules with this particular pharmacophore features. In this particular case, we are looking at 3 acceptors and 1 hydrophobic group. Correct? Zinc Farmer can help you to scan the zinc database and try to find out whether there are any other structure, any other molecules with this particular pharmacophore features. So you may come up with some new novel molecule with that feature. That's the advantage of that. Okay, so we'll go to the second part of it where it's called zinc farmer, which just does the job. Okay, so can you please type this? You should get this screen.
Yes, sir. It's
All of
visible.
you try. Everybody is getting this? Okay. Everybody is getting that? The participants?
Yes, participants. Are you able to see this zinc farmer?
So then you say load features. That feature file which I had sent you, which I hope you have downloaded. Did you all download this from the link I sent you? I hope you have downloaded this. So we have to load that, which contains those 3 acceptors and 1 hydrophobic. Did you see that in PiMode? All of you saw it in PiMode? Okay, somebody answer, yeah. Did anybody see it in PiMode? Those features? Yeah, somebody say yes,
Yes.
no.
Yes. Yes, sir. They have completed up to PiMode.
Yeah, everybody is able to see the features, the 3 acceptors, 1 hydrophobic group. Yeah.
Yes, they are responding. Yes, they are
Yeah.
able to see.
Okay, now you have to upload this thing. We have to upload this file into Zinc Farmer. Okay, open. So I upload this load features. Now you can see the thing, right? Did you all get this? 3 acceptor. Acceptor is in orange color. Now all these 4 molecules are superimposed one on top of another. 3 acceptors, 1 hydrophobic. Are you able to see this? All of you are able to see this?
Yes, sir. Yes, sir. They're able to see.
All of you are able to see this, right? So
Yes.
3 acceptor orange color and then
Yes.
the hydrophobic. Okay, so using the command load feature. So whatever came out from your email, you took that and then you uploaded in the zinc farmer. So all the 4 molecules are superimposed on top of another. Now we are going to search the zinc database. So you have the submit query. Submit. So it will go through a lot of molecules and the zinc database, and then it will identify those which have this type of feature, and it will arrange them in order of RMSD. RMSD means root mean square deviation. Root mean square deviation. It will arrange. It will take time because it is looking at a large number of structures in the zinc database. So it will take time. So it's got lots of molecules. Now let me arrange them in some order. Still can be done. Let me arrange them in some order. Yeah. So this lowest RMSD, lowest RMS, root mean square deviation. So this molecule.
Excuse me, sir. Your volume of voice is very less.
Is it?
Ah.
It's 100%. Abhi, now?
Now it's okay.
This
Now it's okay.
this molecule is the closest to this feature because the lowest RMSD. RMSD means root mean square deviation. So it's identified many molecules and it arranges them in order of RMSD. We will look at each one of them and see. If you click on this, it will go to zinc database and show me what is that molecule. The molecule has a molecular weight of 341.
Yes.
And now you can click on it, all of you, because okay, so it's identifying some molecule in the zinc database. Okay, so it came up with this is the molecule, it says, which has got a closest pharmacophore feature with 3 acceptors, 1, 2, 3, and 1 hydrophobic. So you can see this molecule 1, 2, 3 acceptors, oxygen, nitrogen, and 1 hydrophobic group, right? So this is the closest, lowest RMSD molecule. Some molecule, of course, it's not a drug because, like I said, zinc contains 750 million molecules. So there are only 5,000 drugs. So it contains a lot of structures. Some details of this molecule is given. Log P, then total polar surface area, molecular weight, rotatable bonds, blah, blah, blah. So I want to know some properties. What can I do? So what can I do? I want to know some properties of this molecule. Can
Yes,
somebody
we can,
answer?
yes, sir, we can put that in Swiss admin.
Yeah. So we can put copy the smiles notation.
Smile notation, copy.
Copy it. I want some participants to say because I don't know whether they are sleeping or not.
Participants, please be active.
Yeah. So we go to Swiss ADME and we enter the smiles notation. Okay. And then I say run. So it has picked up large number of molecules, right? It's I picked up around 40,000 molecules with
Actually,
different RMSD values, starting from closest and so on. Actually, so we go to Swiss ADME, and then it gives you a lot of properties. Yeah, you asked me some question.
no, sir, I was answering that actually I have give that option that I have muted all the participants so they can respond through chat box only.
Oh, okay. So it is calculating the property using Swiss ADME. It says GI absorption is high, no blood-brain barrier, inhibitor, good. Then look at the rules. So it's satisfying all the rules. Very good. Log P is about 3.8, quite hydrophobic. Look at the structure. A lot of benzene rings are there. Now nitrogen and oxygen, only 3 chaps are there. Rest all CH2. That's why it's very hydrophobic. 3.5. Average is 3.6. Bioavailability is okay, 0.55. It's satisfying all the rules. Cytochrome inhibitor, yes. Blood-brain barrier, no. So this looks like a nice candidate. So maybe if you do a patent search, this is a novel nobody has reported. If this shows cyclooxygenase 2 activity, maybe it could be even patentable. Who knows? But you have to check the patent databases and so on. Actually, we can look at another other database which I showed you yesterday. Our goal is to get all the properties of molecules and see whether so this is the molecule I have selected, right? So I'm using another database to predict, look at the properties. And not the ADME properties. I use Swiss there. Then I used this software. I use Swiss ADME. So it's running. Okay, so you can try different molecules. Okay, so far we saw one structure. We saw this one, this molecule. It looks a good candidate. Okay, so we can look at another molecule. Like that, we can keep on looking at all the candidates and see the ADME properties using Swiss ADME or using ADME toxicity software. And of course, as a chemist, you will be knowing whether it's easy to synthesize or very difficult to synthesize. That's also very, very important, actually, right? So this is second molecule. Second molecule I clicked on. So it's coming up with another molecule like this. So synthetic feasibility, you as a chemist will be knowing better whether it's possible to synthesize this molecule or not possible to synthesize. As you can see, it contains fluorine, sulfur, and so on. So go to Swiss ADME again. We can enter the SMILES notation. Then you can run. And then again check the rules, various rules, and see whether it satisfies. Yeah. There are some violations. Ghost rule, Lagan rule, violations is there. GA absorption is low. So it's low. Okay, possibly because it's maybe hydrophilic. If you look at log P is high actually 4, but GA absorption comes out low. I don't know why. Blood-brain barrier, no. PGD substrate, no. Moderately soluble, but still GA absorption is low. So molecular weight is 493. It's quite high. So it's done this. It's giving you a lot of information on that. So this molecule does not look very good. That second molecule, because it's got a poor GA absorption. So this has got a poor GA absorption. So you can maybe leave it. So like that, we can go through each one. So please try some just to see how it looks in Swiss ADME and also in this software called PKCSM software. So it's giving a lot of properties here. Okay, this is the second molecule. Doesn't seem to be very great. First molecule looks good in many respects. Okay, so Like that, you can keep going down and down and down. So please do try and see. So you may come up with out of this 49,000, you may come up with some 1,000 molecules which appear to be good. Again, you see a lot of complicated structures. You can run this ADMA.
Sir, please show that slide of flowchart once again.
Yeah, I will. All of you, please try some structures. You must have gone to Pharmacist. Don't just watch. This is not a watching, it's more of a practice. So
Yes.
try something. So this is done the third structure. Again, G absorption is low. Log P is quite high, but bioavailability is okay. Again, some rules are violated. So yeah, but no, so this doesn't seem to be. So we can look at like that. We can go down page 2, page 3. So many. It's come up based on adjusting RMSD. This is the first page contains the one which is closest, and it goes down. So look at so this is slightly more RMSD. Let's look at this structure and see. Yeah, so it's coming up with some other structures with a lot of methyl. Yeah, so this is another structure. G absorption is high. Only one small rule. Rest of is satisfying. Log P has come down. And bioavailability is also quite good. Total polars 93. Acceptors is 5. Ideally, you should get no here. Otherwise, it can get biotransformed. Moderately soluble. Okay, so this is how we keep going down, and we can select, like I said, about 1,000 molecules or something, which looks good based on the commonality of the pharmacophore feature. That's what the whole thing we did actually.
Sir, excuse me, sir.
Yeah.
One participant is asking, please repeat the step of uploading in Zinc Farmer.
Okay, I will do that actually. Yeah, I will do that. So this part is the first part. Okay, where we are trying to identify the common features, pharmacophore features. Okay, so we have to get from SDF file from the Zinc database, photo structures, and then we convert that into mol2 format using OpenBabble. See, sometimes if you are looking for drugs, sometimes they'll give you a commercial name. Commercial name of the drug might not be there in the Zinc. So you have to get the IUPAC name or something like that. You understand? So for example, aspirin has so many commercial names. The people sometimes called Aspro, so many different now. So you might not find the Aspro in Zinc database. So you may have to get the proper name into that. So that's the reason actually. Sometimes you will not find, but if you search, give the proper chemical name, you will always find in the Zinc database. Okay, that is an important point you keep in mind actually. So when you look at go to pharmacist and then get the drug, on top of it they would have written some commercial name. Okay, you might not find, but they would have also written the chemical name somewhere in a corner somewhere. That chemical name only you have to give. But if you go to Google and search for that commercial name, you will get the chemical name. Then load that chemical name into your Zinc database to get the structure. Remember that, because Google will give commercial names, who is manufacturing there, they would have given the chemical name also. So the first part is you get the SDF format, and then you use OpenBabble, convert into MOL2 format, put all the files into one zip file, go to Pharmacist, upload, you will get your Pharma4 features, view them in PiMode. Okay, please practice this. Otherwise, it's not possible, because you have to do step by step many things actually. You can't remember after some time. I am telling you 100%. But once we have the file output from the mol2, that will be also a mol2 format, which we are using for 5mol. The same file we upload in Zinc Farmer. Same file. Okay, that's the same file. Output file. For example, this you can see J41 mol2 file. That's the file which came out from Pharmacist actually. Same file I can view in PiMode. Same file I am going to upload in Pharmacist. Okay. So Zinc Farmer. Yeah, you will get this. Correct. Here you have to say load feature, load. And then that file which you got from Pharmacist output downloaded file, and that you will upload. Yeah. So it is saying no acceptor, acceptor, acceptor. You can read that right on the bottom, and one hydrophobic. If you want, you can remove them also, and then search. So basically, yeah. So it has superimposed all the 4 molecules, and the orange color balls are the 3 acceptors, 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 and there is one green color which is your hydrophobic. This one, the bottom one here. Okay, once okay, so all the molecules are superimposed and you are getting. Then we say submit query. Submit query. Then it will look into Zinc database. So you can see it going round, 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 round. It will come up with structures which have this feature. So it's come up with a lot of structures. I am going to arrange them in RNSD, root, mean, square, deviation. Okay, the one which is closest in superimposable to the one which is farthest. That's what I'm going to upload. Yeah. So this particular molecule has the closest structural overlap. That's why it has got the lowest RNSD. So we can view that molecule, just click, it will go to Zinc and it will show us the structure. Yeah, this is the molecule. 2 acceptors, 3 acceptors, 1, 2, 3, and then hydrophobic, like that, you know. And then what we did, we went to Swiss ADME. Yeah, and then we loaded the smiles notation into Swiss ADME. And then we get the properties. correct? And also we can use the another software which I showed you. Run another. That's called PKCSM. That also gives you. Please provide a smile string. Okay. Then I say I'm not a robot. Then I run ADMT. So I can use whatever softwares I have just to see the properties, whether it's satisfying all the properties. And I think this molecule we saw it is good in many respects, satisfying all rules. Bioavailability is good. GA absorption is also good. Log P about 3.8. Quite hydrophobic because lots of benzene rings only in minus. We have two oxygens with nitrogen. So lot of CH2s and aryl groups. Solubility also moderately soluble. But this looks like a good candidate. So as a chemist, you can see whether this can be synthesized. So this could be having the cyclooxygenase 2 inhibiting property because that's what we saw initially, right? Selective cyclooxygenase 2 inhibitor. So hopefully this also may be a selective cyclooxygenase 2 inhibitor. So this could be, if you search all the patent databases and if it looks like a novel molecule, if you do a biological activity and it shows activity, maybe it's a novel molecule. It has not been reported by anybody. And it will have the ADME and drug likeness property because we checked with the Swiss ADME and we also checked with other softwares also, right? So hopefully it can become a drug also because it's satisfying all the conditions per se, drug likeness conditions. Okay, any doubts? Zinc Farmer cannot be downloaded. It's a server, so you have to upload. Okay. Somebody asked. Zinc Farmer, it is not a downloadable file. Yeah, Zinc Farmer is you upload. Let me show once more. No, Zinc Farmer is a server, online server. Oh, Zinc Farmer is an online server. Everything is online. Zinc, it's an online server. Zinc Farmer is a free. Yeah, it's an online server. Then you load feature. Your output that came from a Pharmacist, which you downloaded, okay, has to be uploaded. Any doubts? Somebody having problem? Is it? There is no uploading.
Yes,
uploading, you have to say load feature.
Load features. Dr. Sukhvinder, you have to go on load features.
You have to load feature. You upload your downloaded file from the previous activity, which came in email. You clicked on it. Then there must be a file that must have come .mol2 file. So that you save it on your desktop, wherever it is. Then you say load feature. Okay,
Yes,
load feature.
participants are able to upload.
Yeah, so this
there
is
is
the file. Okay. You have to load feature. Yeah, abhi aa gaya na. So you load feature. Okay, so this has got 3 1, 2, 3 acceptors, 1 hydrophobic. And then you say submit query. That time, if you look for similar pharmaceutical featured molecules,
Dora Babu is asking, can you please demonstrate complete process with some other drug molecule? It's
Yeah.
better you practice first, Dora Babu.
Yeah, that is why I gave half an hour time. If because it's very important. I will show you, but it's very, very important that you practice. This cannot be remembered because this is not like a formula or something unless I practice. I will show you, but unless you practice, you make mistakes, ask doubts. It's very difficult. I can show another molecule, but the same procedure only. I will show you that definitely. You have to practice, not just watch. No problem, even if you miss out once or twice mistakes automatically. But unfortunately, you are alone, so it becomes difficult. In a classroom, you can ask your neighbor.
Excuse me, sir.
Yeah.
Actually, a participant is saying that when they open this zinc farmer, they are not able to get these features, add feature, load receptor. ये वाला उनके पास नहीं आ रहा sir.
That what can I do? I can't do anything. No. So because it's automatically will open in Windows. I don't know what they are using. Are they using Apple or something?
Okay,
are they
okay.
using Mac or something? Mac. So Mac because most of these programs run on Windows. So I don't know Windows. These are all window-based programs.
Okay, okay. Some participants they're saying it's available. Maybe then Dr. Sukhwinder, you please check it once again.
Yeah, because many of these software they all are window-based. You have to read the commands. I mean the front page. In many cases to see, because nowadays many softwares for Windows download, for Mac you download like that double time. So
right.
You have to very carefully read that. Okay. But many of them are window based, mostly default window based default. Of course, there are some Mac based also, but default is window based. So you have to read properly and see what is. Okay. Please practice, otherwise it becomes difficult. It's not like theory. Now, I can show another molecule also. Of course, I'm going to show you that. Okay, there are many drugs available for lowering blood pressure. One of the classes are called ACE inhibitors, angiotensin converting enzyme inhibitors. Angiotensin is an enzyme, angiotensin enzyme, ACE enzyme. It converts angiotensin 1 to angiotensin 2, which is involved in vasoconstriction, leading to increased blood pressure. So there are many drugs trying to block this ACE actually. You must have been hearing about ACE2, right? Recently with this pandemic. That's another enzyme, angiotensin 2, that converts angiotensin 2 to angiotensin 1 to 7. In fact, that enzyme is very important, ACE2. In fact, your spike protein in SARS-CoV-2 enters the host through that ACE2. Okay? And the ACE2 is an important enzyme which converts angiotensin 2 to angiotensin 1 to 7. Thereby, it's not allowing accumulation of angiotensin 2, which is a vasoconstricting system. So you must have been hearing about ACE2 in the past 1 year. But our focus is on this ACE, angiotensin converting enzyme, which converts ACE1 to ACE2. So there are some drugs which are called ACE inhibitors to control blood pressure. Okay, they're all called captopril, enalapril, zofinopril, ramipril, fosinopril. Lots of drugs are there, which are controlling blood pressure by blocking the ACE enzyme, angiotensin converting enzyme, which converts ACE1 to angiotensin 1 to angiotensin 2. Okay, same procedure we will go through. Okay, if you people have interest, you can also try. Go to captopril, go to zinc database. Zinc database. Please try captopril. Captopril, yeah, it's there. So I can. I'm going to type each one of them. Sorry. I'm going to same way. I'm going to take all these five, get the SDF format, and then convert it to MOL2 format, and so on. Right. So captopril is there. Search. So it's a slow process, but you have to do that. No other way. This captopril, I can download the SDF format, right? Please try. When I am doing also, you can also try. Just don't watch, because it's not a watching show. Okay. Then we go to next chap is enalapril. Enalapril. ENAL April. First is captopril. You can write it down. You can also try enalapril. Enalapril. They have enalapril here. Yeah, this is enalapril. Download the SDF format. I'm all saving them together. Save link as. I'm all saving them together in one folder called them2. We can also enalapril. Okay. Then we go to third molecule, zofinopril. Zofinopril is a OFENO-pril. Zofino. Zofinopril. I don't know. Rated zinc database. In zofinopril. Fourth molecule, fosinopril, FOSIN. Okay. Then next molecule is 5 molecules we are looking at. Fosinopril. Then last one is ramipril. RAMI. Ramipril. Okay, so I have done the first part. Now I need to convert each chapter one at a time. Yeah. So I have got all the 5 molecules: captopril, enalapril, fosinopril, ramipril, zofinopril. Now I have to convert them all into mol2 format. So I go to OpenBabble, MTF to mol2. So just check that, and then go here. I go to desktop, CentOS, so captopril, open. So it's come out with the structure. Captopril. I'll go one at a time. So I converted, then I go to fosinopril. Then I go to fosinopril, fourth one, ramipril. Then comes fifth one, zofinopril. So I got 5 files. I don't think I found it. Captopril and, right. So hopefully it should work. So everybody got this, so it's running. If there is no error, that means it's running. It says results will be sent. Any doubts?
Now I think it should be clear to all.
So if there is any error in your, if there is
Dora
any,
Babu was asking.
if there is any error, if there is any error in my file conversion, something there, it will give an error and it will just come out. Now it's gone to my, hopefully, it should come. Oh, again, you can view your result under this. So, no, this is old one. So it's still not, it is not come actually. Oh, I think, thank you, 531. You can view results in the, it's come. So it's come here. Sometimes it comes fast, sometimes it comes slow. No problem. So what has happened? Lopinoprid, enalapril, ramipril, captopril, fosinopril. So it's given something actually. Okay, so let us see how it comes out. PiMode. So 4 is, so it's come up with the okay, it's come up with 4 features. Label, atom name. So 2 hydrophobic, 2 acceptors. 2 acceptors, 2 hydrophobic groups. We can calculate distances. Measurement. 3.1. So we can gives you distance. So if you look at the molecules, two hydrophobic, two acceptors. You can see two acceptors, maybe two hydrophobic. That's the common feature of this. Two acceptors, two hydrophobic. So two acceptors, that means two oxygens, you can see, no? And two hydrophobic, a lot of hydrophobic. So same story, we can go. Yeah, two acceptors, two hydrophobic groups are there with this distance. So we can again go to zinc of farmer to find out there are any new molecules with this feature. Load feature. Two hydrophobic green color, two acceptor orange color. So even the downstairs, down you can see two hydrophobic groups. Now, yeah, you can see two acceptors. Now you can say submit query. You can see two green balls and two orange balls. Okay, now you can say submit query. So you will get a lot of new molecules with this feature. You can do this with ADME. Yeah, so it came up with lots of colors. Yeah, this is the closest. Structurally, pharmacophore wise, this is the closest acceptors to hydrophobic. Is it easy to synthesize? There are a lot of chemists in your group. Do you think this is easy to synthesize? Eight-member ring. Anybody?
Yes, participants, please respond.
Yeah, is it easy to synthesize or it's difficult to synthesize? Is it difficult? Eight-member ring is difficult to synthesize. There is a nitrogen eight-member ring because there are so many of you chemists in that group. Okay, so this looks like a simple looking molecule. It's satisfying many things, pharmacophore features, but bad luck. Maybe the chemist will say we can't synthesize, right? So it's got all the features of pharmacophore features, but maybe it's difficult to synthesize. Eight-member ring,
Generally, 5 or 6 member ring is stable.
6-member ring with nitrogen is okay, like pyridine like. But then this is a 8-member ring, so problem. Okay. So we ditch this. You may go into some other structure. And so we like that we have to keep looking. It is not only properties, but as a chemist, you will know better whether it's easy to synthesize, will it be stable, all those things, right? Look at this. More complicated. Bicycle. Difficult, right? This one difficult, easy. Synthetic feasibility also has to be seen. How easy to synthesize. So computer will give out a lot of new structures based on the rules, but how easy it is to synthesize, you know better.
Yes, participants, they are responding that bridge compounds, they are difficult to synthesize.
Yeah, fish compounds. You can give it as a PhD project with lots of
Yes.
substituents and all that, right? Maybe that's why these are the drugs as ACE inhibitors, right? These are much easier to synthesize, right? Yeah, 5-member nitrogens are there. Then some fused rings. Okay, see, so we saw today pharmacophore-based approach. So I request you practice. I will send you the structures if you want, both the COX2 as well as this, and try the entire process and see whether you get something different answer or same answer. So that way, yeah.
So actually, there was one query from the participant that the link which was shared by you today regarding that build QSAR.
Yeah.
So they are facing again problem in that link. So
Yeah,
they
no.
are asking that whether tomorrow session is based on that QSAR. So that link is
XAR is one of the softwares I'll be using. Why? What is their problem?
yes.
They are not able to open the files. Can somebody else see whether they are able to open the files?
Files to open हो रही है sir.
Then you have to.
Dr.
Then you have to.
Sukhvinder,
You have to.
Dr. Sukhvinder, you
You have.
please tell.
Please copy it into your program.
Okay.
Okay, into your C drive in the program files, and then you set up the software.
Okay, sir.
Take it to programs, file in C drive, and then try to run
Okay,
it.
sir. Okay.
Yeah.
ठीक है, that is clear to the participant. You copy those files, and then you will be able to use that software.
Yeah, because all the files, everything is there.
Today's session was very much interesting. That any query? Yes, participants, any query?
Yeah, tomorrow we will look at QSAR. That software is only one part because there are other so many things. QSAR is not just one software.
Sir, molecular docking part that will take on 4th day only.
Yeah. Yes.
So tomorrow's session is based on QSAR.
Yeah, fourth day will be target-based design, where you have the homology modeling and docking,
Okay.
and then looking at target prediction and so on.
So I think today's session was very much interesting. Participants are very happy, and they get the complete information. And a feedback link has been shared in chat box.
Okay.
Yes, participants, feedback link has been shared in chat box. We will keep it open for 5 to 10 minutes.
Okay, tomorrow we will meet. We'll
Yes.
talk about quantitative structure activity relationship and what are the various features in QSAR. Okay.
Okay. Thank you, sir.
Thank you very much.
Chandra, you want to say something? Chalega.